Most of the world didn't have it until colonization started in the 15th century.
There is something flowing through the veins of millions of people right now, something that scientists have spent decades trying to fully understand, and something that, the more they study it, the more it refuses to give up its secrets. It is not a drug, not a therapy, not a technology. It is simply a blood type. Type O. The one marked on emergency room charts. The one doctors reach for first in a crisis. The one labeled universal donor in every medical textbook ever printed. We think we know it. We have been told it is straightforward biology, a matter of antigens, or more precisely, the absence of them. But what researchers have quietly uncovered over the last 2 decades paints a picture so extraordinary, so deeply strange, that it changes the way we must think about human survival, ancient catastrophe, and the hidden code inside our own bodies. This is not a story about blood transfusions. This is a story about what it took for the human race to survive. When population geneticists first began systematically mapping blood types across the globe, they expected to find roughly what they had always assumed: a mosaic of A, B, AB, and O scattered across different regions, shaped by the random movements of ancient peoples. What they found instead, particularly when they turned their attention to the indigenous populations of the Americas, stopped them cold. In community after community, stretching from the Arctic coast of northern Canada all the way down through Central America and into the deep interior of South America, type O blood was not just common. It was, in many places, the only blood type present. Certain Amazonian tribes showed type O frequencies approaching 100%. Andean highland communities, separated by mountains and centuries from lowland populations, showed the same extraordinary pattern. Indigenous groups in the Great Plains of North America, coastal peoples of the Pacific Northwest, remote Patagonian communities at the southern tip of the continent, all of them told the same story. Type O, and almost nothing else. The initial scientific instinct was to reach for a familiar explanation: genetic drift. In small, isolated populations, random chance can cause certain traits to become dominant simply because those early founders happened to carry them. If the first humans to settle a region were mostly type O, then over time, that trait could come to dominate. It was a reasonable hypothesis. The problem was that the data soon made it unreasonable. Genetic drift produces uneven, patchy results. It leaves behind traces, pockets of variation, occasional outliers, communities where A or B managed to persist simply because of chance. But what researchers found across the Americas was not patchy. It was sweeping. It was consistent in a way that random processes almost never produce. In many populations, types A and B were not merely rare. They were completely absent. No traces, no remnants, no survivors. It was as though they had been erased, not gradually, not randomly, but deliberately and completely. That word, deliberately, made scientists deeply uncomfortable, because if it was not random, then something had selected for type O. Something had, over time, removed the other types from the gene pool. And whatever that something was, it had been powerful enough to accomplish this across an entire continent. For a long time, researchers lacked the tools to look back far enough to understand when this transformation happened. That changed with the revolution in ancient DNA analysis. By the early 21st century, geneticists had developed methods capable of extracting usable genetic material from skeletal remains tens of thousands of years old. And when they turned those methods toward the question of blood types in early human populations, the results shattered long-held assumptions. Studies of ancient human remains from Siberia, the region from which the first Americans are generally believed to have originated, showed something crucial. These ancient Siberians were not genetically uniform. They carried a diverse range of blood types. A, B, and O all appeared in significant numbers, much as they do today in populations across Europe, Asia, and Africa. This is important because it tells us that whoever crossed from Siberia into the Americas during the last ice age did not arrive carrying only type O. They brought a full spectrum of blood type diversity with them. And yet, somewhere in the Americas, that diversity disappeared. Analysis of ancient human remains from North America, dating back thousands of years, showed type O present in the earliest layers of settlement, and then, moving south and forward through time, it vanished. By the time the record reached the Central Americas and the Andes, A and B were gone. Type O alone remained. This was not gradual. It was not the slow fading that drift produces over many thousands of years. By the standards of evolutionary time, it looked rapid, directional, purposeful. Researchers began to use a term that carries enormous weight in population genetics, a bottleneck event. Something catastrophic had struck these populations, something that killed enormous numbers of people. And critically, it had not killed them randomly. It had killed them in a way that left Type O carriers standing while A and B carriers perished. The most compelling explanation, and the one that has gained the most traction in the scientific literature, is disease. Specific pathogens are known to interact with blood group antigens, the molecular flags that sit on the surface of red blood cells and define your blood type. Some pathogens use these antigens as docking points, binding to them to gain entry into the body's cells. If an ancient epidemic was caused by a pathogen that preferentially bound to A or B antigens, then individuals carrying those blood types would have faced a severe immunological disadvantage. Type O individuals lacking those antigens would have had a meaningful survival edge, not immunity, but enough of an advantage that, across many generations and many outbreaks, more of them survived to have children. Over centuries, that small survival edge would have compounded. Each epidemic would have further thinned the ranks of A and B carriers. Each generation would have seen Type O become more dominant. Eventually, in the most severely affected populations, A and B would have disappeared entirely, their carriers having died out long before modern history began. The disease hypothesis gained powerful support from a completely different direction: modern immunological research. When scientists began systematically comparing health outcomes across blood types in large population studies, they found a pattern that was subtle but undeniable. People with Type O blood, on average, showed lower rates of severe illness and death from several historically devastating diseases: cholera, certain forms of malaria, and specific hemorrhagic fevers among them. This is not to say Type O is a shield. Type O individuals get sick. They die from infectious disease. But when researchers measured outcomes across thousands and tens of thousands of patients, the survival odds tilted, just slightly but measurably, in favor of Type O. In a world without medicine, without antibiotics, without vaccines, in the world of 10,000 years ago, that slight tilt would have been the difference between lineages that survived and lineages that did not. Adding another layer to this, researchers studying indigenous communities in the high Andes of South America discovered an unusual concentration of a specific immune gene variant known as IL-17 variant 52C. This variant affects the regulation of inflammation. One of the primary ways that severe infectious disease kills is not through direct damage by the pathogen itself, but through the immune system's overreaction to that pathogen, a cascade of inflammatory signals that, when unchecked, destroys the body's own tissues. The IL-1752C variant appears to help regulate this response, keeping inflammation strong enough to fight infection, but controlled enough to avoid catastrophic self-damage. What caught researchers' attention was that this variant appeared far more frequently in people with Type O blood than in the general population. It was not a universal feature of all Type O carriers, but the association was strong enough to suggest that the two traits had been shaped by the same selective pressures, that whatever ancient catastrophe had pushed Type O to dominance had also, at the same time, favored individuals whose immune systems were better calibrated to survive it. In 2024, archaeologists working near a remote lake system in Siberia recovered a find that would add yet another dimension to this story. Preserved in permafrost for more than 12,000 years, the skeletal remains of an ancient individual were extracted and sent for genetic analysis. The condition of the DNA was remarkable, detailed sequencing was possible, and what it revealed was extraordinary. Within the ABO blood group gene cluster, researchers identified what is known as a deletion mutation, not a change in the genetic code, but an absence. A segment of DNA was simply missing. Deletion mutations of this kind are significant because they are not the product of ordinary genetic variation. They indicate that, at some point, something exerted extreme pressure on this lineage's biology, forcing an adaptation drastic enough to involve the loss of genetic material rather than the simple substitution. This mutation, designated by researchers as a unique O-type variant, was absent from European DNA. It was absent from African and East Asian samples. It was even absent from most other Siberian ancient remains, but it appeared in the genetic profiles of specific indigenous North American populations, drawing an unbroken biological line from that 12,000-year-old individual in Siberia to living communities on the other side of the world. It was, in the language of genetics, a founding signature, the mark of a small group of survivors whose descendants would go on to populate an entire continent. The implications were sobering. This mutation had not appeared by accident. It had been forged by pressure. Whatever this ancient individual and their community had faced, whether a disease, a toxin, a multi-year catastrophe, it had left a permanent mark in their DNA, one that their descendants still carry today. As if the story were not already complex enough, recent genetic findings have extended it across an ocean. Skeletal remains recovered from Rapa Nui, Easter Island, dating back approximately 900 years, were submitted for blood type and immune gene analysis. The results connected two worlds that had long been thought separate. The remains showed a high prevalence of type O blood. That alone was not surprising, given the island's Polynesian heritage. But researchers also found the IL-1752C immune variant, the same variant identified in highland Andean communities in South America, thousands of miles across open ocean. This variant is rare enough that its presence in both locations almost certainly indicates direct genetic exchange rather than coincidence. It points to contact between Polynesian voyagers and South American populations centuries before any European arrived in the region. The significance of this connection goes beyond historical curiosity. If Polynesian navigators carried not only goods and cultural knowledge, but also specific immune genetic adaptations shaped by their own survival trials across the vast Pacific, then the spread of Type O survival toolkit was not limited to land migrations. It traveled by sea. It was carried intentionally, in the bodies of explorers and traders and alliance builders, from one side of the world to another. What began as a medical footnote, a blood type notable mainly for its usefulness in emergency transfusions, has become one of the most remarkable stories in the history of human genetics. Type O blood is not simply an absence of antigens. It is the product of trials that most of humanity did not survive. It carries, encoded in its very structure, the memory of epidemics we have no other record of catastrophes that erased entire branches of the human family tree and left only those with the right biological tools standing. Every person with Type O blood is, in a sense, the descendant of survivors, not survivors of one event, but of many, of plagues that swept ancient continents, of bottlenecks that compressed entire populations down to a handful of individuals, of migrations across ice and ocean that tested the limits of human endurance. Their ancestors made it through, and the evidence of how they made it through is still written in the blood. The science is not finished. There's much that remains unknown. What specific pathogens drove the ancient selection? How many bottleneck events occurred? Whether the hidden genetic sequences researchers are only now beginning to identify have functions we have yet to understand. But with each new discovery, the picture becomes clearer. Type O blood is not just a clinical category. It is a living record of what the human species endured and survived. And if your blood carries this ancient signature, it means the most resilient line of human survival runs directly through you. Like, share, and subscribe for more deep explorations of science and history. And watch the next video for more hidden truths buried in our biology.